Yeah. It depends.
I need to, I'll say to myself.
Alrighty, we're live. Hi, everyone. Welcome to exam one review. This TV sucks. Anyways,
But it's really good. It's fine. Yeah, it's nice.
What's wrong with the TV?
everything.
You're a complainer.
Depends on the projector. I can be a hater. Okay, so we're gonna start. We wanted to switch the order because I thought that intuitively protein stuff comes first and then CML stuff can come after. And it's easier to understand the CML stuff because of the protein stuff. So I decided to switch the orders. But so we're gonna talk a little bit about protein misfolding. I promise you I will not go over the 20-page freaking hallmarks paper. I will spare both of us, you guys and us, of this. And so we will cover the hallmarks, but we won't go into like 20 pages worth of detail, thankfully. Okay. So I don't like that box. Hold on. Alrighty. So first off, we gotta talk about structure of proteins. So we have primary, secondary, tertiary, quaternary. Primary is just the sequence of amino acids in a polypeptide chain, and for each amino acid to be added to the chain of other amino acids, it needs to go through a dehydration reaction. And it gets added onto the C-terminal end of the amino acid chain. Secondary structure are the structures such as alpha helices, beta sheets, and beta helices that exist. And these are formed with the interactions of the atoms in the backbone of the polypeptide chain. So these interactions are typically non-covalent, and they are oftentimes polar. And here we also have tertiary structure, which is the 3D folded structure of the polypeptide chains. And important to note here is that these interactions, in order for the tertiary structure to be formed, are happening between R groups of the different amino acids. Most of these, as we were talking about, are nonpolar. So you have your dipole bonds, you have your ionic bonds, you have your ion dipole bonds, and then the strongest out of all of the bonds between R groups is the disulfide bridges formed by two cysteines, and this is because this is a covalent bond, and so it is stronger than your other non-covalent polar interactions. Any questions so far? Yes, no, maybe so? No? Okay, and then quaternary, for quaternary structure to happen, you need multiple polypeptide chains to come together and form three-dimensionally into one protein. But these are gonna be different polypeptide chains that are going to three-dimensionally fold and then come in together as one protein. An example of that is hemoglobin, antibody. There are plenty of examples. Okay. You want to get this one?
Yeah. All right, so protein folding is like this multi-step process that starts from this unfolded protein. Eventually you end up with the finally folded form of it. But it goes through many intermediates, and it generally does that by seeking the lowest energy form. So a protein starts out in this unfolded state, like up there, that's the highest energy it could be. And then it will naturally, by like the order of the universe, go down in energy and seek that most stable form. And in between that, it's going to hit intermediates that can be influenced by these little energy humps that dictate, like how it's going to enter that low energy state, or chaperones that can kind of force it to fold a certain way and end up in that native state, which is, the preferred way that we want proteins, because that's when they function properly. These chaperones sometimes can not function correctly. That's what's dictated by that little red, dash line. And when those chaperones aren't there, that's when you can sort of end up from your like partially folded intermediate state to something you don't want. Like that's when the aggregates form, the oligomers form, and you get your fibrils. And if you notice something, the fibrils in this image are much lower of an energy form than a native state, which kind of speaks to their stability. So later when we kind of talk about, you know, combating these fibrils when they form and how we can destabilize them. I think Laura was talking about like the high temp, high pressure method of like, you know, dealing with these kind of things. This is why these are so resistant. Very low energy, very stable. And then it's also looking for my last review. So I'm not sure. Yeah, let's see. Oh, and proteins generally fold in units of the secondary structure that John was talking about. So those are your beta sheets, the alpha helices. And, you know, we talked about this in class a little. The beta sheets are most relevant to our fibrils because those are the ones that form those like really strong interactions, those covalent bonds, and you have way more surface area that you can pack close together. And that's what kind of helps it hit that low energy state, which is the most stable. What else? Yeah, I think and so I guess we can start talking about the most important, I think, part of this unit is these hallmarks. Although it's a really long paper, these are pretty important. So we'll go through each of these. Stop us if you have questions on any of them, because I know there's a lot. You want to take the first one, John?
Sure. So first one is the pathological protein aggregation. So this was first found as a pathological agent in prion disease. And so these are aggregates that form once a protein is in a misfolded state and it goes and is attracted to other proteins of its kind that are fully functional. And it leads into a misfold of the other proteins that are functional, which is how these are going to cause larger and larger misfold events for happening. And so these are toxic because we were talking about in class, once a protein has its own three-dimensional structure, it is supposed to form or is supposed to perform its own function. And if it is out of its main structure type, then it will not be able to perform its function. And as we have seen with, especially with these neurodegenerative diseases, once a protein is out of its own structure normally, it'll go on and aggregate, and this aggregation will cause further aggregation of more proteins that are functioning normally. And this comes into the theory of seeding, which is when these oligomers of proteins that are misfolded break off from the larger misfolded structure and go into completely different parts of the cell, and then start again to recruit these proteins in natural state. And then the process of oligomerization and seeding restarts. And as you can think, if one of these proteins is being shipped off and it's recruiting more proteins, it's a very, very fast process that can be very, very much toxic to the cell. And here, just a little bit of how the cell tries to protect itself from this from happening. So you have proteasomes that are going to be degrading smaller aggregations. And so you have processes of ubiquitin-mediated proteolysis that happens in the proteasomes. And you also have your lysosomes, which are known as like the trash can of the cell. And these are going to be degrading the larger compounds. Don't know exactly the process that happens inside the lysosome. I don't think a lot of people do. But you just need to know that it has a higher capacity of degradation than the proteasomes do. But the lysosomes specifically happen to perform this function of being like trash cans of the cell for like a bunch of different functionalities. And it's also important to determine that the proteasomes are actively, like they're constantly working with degradation for smaller compounds because ubiquitin mediated proteolysis is happening like all over the cell as a means of regulation for a bunch of cell processes. And something you have to keep in mind is that once you have all these protein aggregations happening and the cell is going crazy trying to get rid of them, that can also affect the proteolysis that's supposed to happen for controlling other processes in the cell. So that can cause stress and that can cause malfunction of other functionalities. Good? Yeah. All right.
Okay, so our second one is a synaptic and neuronal neuronal network defects. So this I like to think of as kind of like, remember that weird, like really big and convoluted chart that Laura put up? And it was like, this is like the end stage of that, right? So everything that happens with the aggregation and oligomerization or whatever, and all those defects in the chaperones, this is generally what you end up with. And because of that, this is where you're going to see the phenotypes of the diseases show up, right? So we have any sort of like dysfunctioning protein or aggregation or, anything like that will lead to can lead to synaptic failure. This is the early event before you hit your NDD. So this can include things like changed energy metabolism, right? Neurons are they do a high rate of metabolism. They need a lot of energy. If you disrupt that, you're going to have defects. Oxidative stress is a big one. That just causes havoc everywhere. I mean, I think in 285, we talked a little about how that leads to damage in DNA, but you can also just damage cellular function, especially when you need to transmit signals at a rhythmic and consistent basis. Protein production and degradation is disrupted. This leads to the buildup. I think that's pretty self-explanatory. Cytoskeletal dynamics are important, and we'll talk a little bit more about that with the fourth hallmark. And then eventually all these things, if the neuron can't work right, it can't signal right, and it can't, sustain the energy it needs to function at all, then it will lead to death. And that's when you kind of see, the main phenotypical features of, let's say, like Alzheimer's or ALS or something like that. Right? Anything else? Yeah.
Okay. So third hallmark is aberrant proteostasis. So this happens with the malfunction specifically, and it's a more broad hallmark of the malfunction of proteins because they're not in their optimal state for functionality. And so this can happen via premature termination. This can happen with wrong mediated proteolysis of specific proteins. This can happen because there is a missense mutation in the protein sequence that causes the protein to be smaller or larger. It causes the amino acid sequence to be different. And even if it's a single point shift mutation, that can alter the frame, that can alter the amino acids that are being synthesized, and that can also alter the total length of the protein. So if you think about if you're switching off one amino acid in the stop codon, and all of a sudden it's no longer a stop codon, you're going to have to rely on luck for the next stop codon, however many bases down to be, for you to have the protein terminated. And so, as Laura's main 285 point is, protein structure will lead to its function. And so if you are altering this functionality, if you're altering the structure in any way, shape, or form, then you're ultimately going to affect its capability of performing its function. So aberrant proteostasis has to do with the maintenance of these proteins, or the lack thereof maintenance of these proteins in their most optimal state for performance. And so it just leads to a lack of proper functionality of individual protein function and also systematically. Okay.
Okay, so the fourth one is the cytoskeletal abnormalities. So this kind of ties into the neuron stuff I was talking about earlier. But basically you have three important forms of your cytoskeletal system that we talked about in the papers. These include your tubulin-based microtubules, intermediate filaments, or the neurofilaments, and the actin-based microfilaments. And the cytoskeleton is essential to transporting very important information and cargo, especially considering the neuron. The neuron is a bigger cell than most, right? It's really long, and it serves a very important function. So having the cytoskeletal system is kind of essential. You need the rigidity in the cell to maintain its form and those connections between the synapses, and to just establishing like gradients, for example, right? If you can't establish the right gradients between, the ions, then you're not going to get a signal that transmits correctly. And this is the one that may interact the most with other NDD hallmarks, especially considering aggregation, right? The cytoskeletal system in the normal cell can function in like finding misfolded proteins, transporting to where they need to go, degrading them, recycling them, whatever. If this isn't working right, then you're not fixing the root problem when it arises. Right, like we talked about, I think Laura mentioned, like misfolded proteins happen all the time. Like that's just normal, but we deal with it efficiently in other parts of our body, and that's why we don't have problems. If you can't deal with it correctly, then it's kind of impossible to recover, right? Once they start aggregating and those seeds form or whatever.
Your question that relates to the problem set, and like, that's why do we think that smaller seeds would be more toxic than larger aggregates? Is that because they can like travel more?
I think it's because, and John can check me on this, but when you have a smaller seed that breaks off, it's kind of easier for it to like go around, collect and bump into other things. And two, it doesn't kind of get stuck. Like when you have the really long seeds in like those kind of structures, I mean, you got to think that yes, they're bigger, but they're also going to move slower and they're going to get stuck around in areas or the cell, right? When you have smaller seeds, it's much easier to get around, build interactions with other little small seeds and then aggregate that way.
I also think that I would tackle that by thinking about how just overall a larger protein is easier targeted or is easier to target, or not a protein, but an aggregation of proteins is an easier target for any cell misfold or unfolded protein response to find. Whereas a smaller, just a seed can go really fast wherever into other parts of the cell, and it doesn't happen to be quite as responsive to like all the other unfolded protein response, sharp response, whatever. Because a lot of these cases of protein misfolding are happening because of like outside factors, that being pH of the cells, that being like heat, and that being like less so in the neurons for like sun and photons and everything like that, but more so that the seeds can travel faster and they're harder targets for mediated responses to occur. Okay, so altered energy homeostasis. A little bit of this goes along with what Aaron was talking about earlier. So synaptic energy of the neurons is a high energy consuming event. And think about not only the neuronal network happens to be very large, but you're also not going to end up finding the target every time you fire. And so these cells are very reliant on a very well-rounded response to their use of energy with a higher than normal presence of mitochondria. And so as we learned from we were younger and even later on when we actually like studied glycolysis and the electron transfer chain and the proton gradient, mitochondria are very important for the production of ATP. And so if you think about the neuronal cells having a lesser availability of ATP, it doesn't, not necessarily, in this case, not necessarily because of any faulty procedure, but just if you're using too much energy, you will be in mitochondrial stress response, and you'll also not have enough ATP to go around and perform as many neuronal connections as you need. But if this happens to be because of a malfunction in mitochondria, so like a genetic mitochondrial disease, or like, for example, Friedreich's ataxia, where mitochondria have this malfunction just naturally, then the neuronal cells do not have this energy source that they can utilize and perform their functionality. And as we know, most of our cognitive function, our sensory function, our motor function happens because of our neuronal network. And so if you have low ATP availability, that is just not going to be optimal. And so mitochondrial dysfunction can also lead to oxidative distress. It can lead to just damage overall. If your brain also cannot perform oxidative electron transfer chain mechanisms, then just through hypoxia, you also run out of energy. And this ultimately can lead to your neuronal network having higher than normal cell death. Yes, Jen?
Is the high energy demand of the brain causing mitochondrial dysfunction? Or
in this case, it's more through like for that image that we're looking at, it's more through like the use of energy for functionality, so physical activity or just like any sort of use of the brain, like in like when you take a test or when you're performing like a high quantity, like high stakes thing. But these generally, when you have like a mitochondrial dysfunction that's not caused by that, it's like a genetic malfunction of the mitochondrial DNA.
And I know, like, I've seen this to you before, but I feel like I still don't really get it, like, they say, like, use your brain, keep your brain healthy, like, do these puzzles and these complicated things with your brain to not get MDBs. Isn't that counterintuitive advice?
And I mean, I like to tackle that by thinking of the brain as like any other muscle, and that just you have a natural necessity of usage for it. Again, we're not talking about being like triathletes of the brain here. We're not talking about like, over stimulation of like your muscles because you're an athlete, but just general cognitive tasks. Once you stop using them, like your neural network doesn't necessarily need to be as active. And again, I'm not like a neuroscience expert here. But push comes to shove, if you don't use one of your muscles, like if you're used to going to the gym a lot and bench pressing a lot, once you stop going to the gym, you won't be bench pressing as much weight as before. And that's just because you're not utilizing your muscles and your muscle fibers. Now, do the cells of the brain work exactly as other muscle fibers? No. And does the neuronal network have a lot to do with that? Yes. But I think of it as the brain being storage for the neural network to work. And you have different parts of the brain that are working. So your cerebellum is working different than like your frontal lobe, and so it goes. But I think that you can think about it as you're stimulating the brain in a way that causes it to maintain its functionality rather than like not performing high intensity like cognitive functionality that leads to you not using it. I mean, you're still gonna, do your daily tasks and perform cognitive function, but it's one thing if you're actively like engaging, reading, performing, like doing math, and then just like standing around and doing nothing all day. Hope that helps. I hope it didn't confuse you more. But yeah, so that's homework 5. Any questions? You have a question, Neha?
All right.
Oh, sorry, Grace had a question. Yes, Grace.
My bad. Could you
Yes,
give
you're bad.
could you give an example of a function that may not happen as a result of like too much energy usage that could contribute to like MPDs, like if there's like an over usage of like that?
Like ATP wise?
I guess so. I don't know. I just like wrote down here, like if there's like too much energy usage, there could be like a mitochondrial stress response and not enough ATPs to go around and perform functions. So like what are some of those functions that may like
So if your brain-
perform?
So if your brain is under hypoxia, right? The electron transport chain is aerobic method that's occurring inside of your cells, right? And so in theory, if you're under hypoxic stress, like you're trying to use the least amount of oxygen possible and you're trying to like reduce your functionality. So you could argue that if you're running out of oxygen, you're not performing the like regeneration of ATP molecules as strongly. And then that is affecting the brain usage and like the neuronal cells activation of energy. I don't know an exact like answer to your question. I'm just like bringing together like what I can think of as far as like you asking that question. But I don't really think like a specific thing would be like that.
Okay, but like I just don't understand really like why it's like a hallmark then, because like things aren't being done that need to be done.
Why it's a hallmark?
Yeah, like.
Like altered
energy
mitochondrial function?
Altered energy homeostasis, yeah.
Well, I mean, if you're constantly having less energy available, then by default, you're constantly gonna have a lesser activation of your neuronal network, and by default, that also leads to like a smaller performance and like neural connectivity and stuff like that.
Okay, thank you.
Yeah. I could also add that, like, I think Laura was talking about this, like when the example with having, the kids that require a lot of input and the ones that don't. If you for example, like have a certain set limit of energy that now you have to divert to helping, this dysfunction and fixing that. Then the ones that normally work are now suffering from this altered homeostasis and like they're not getting the sustained energy that they should. And that's why you're seeing that hallmark pop up. Like the things that should be working right aren't because now we're diverting our resources to somewhere else. Yeah. We good?
Also, thank you guys for on the way.
Yeah, I let her in. Thanks, Dasha. So, hi, Dasha. DNA and RNA defects, are sixth hallmark. So, I mean, this is kind of expected, right? Whenever you have cells under stress, low ATP, your repair mechanisms aren't working correctly because, like I said, one, you might not have the proteins available to do it. Two, you're diverting resources, and these things are, I wouldn't say high energy processes, but they're not low, right? They require some of this like cell resources to be like, hey, we need to go here and fix this, and then we're gonna focus on our normal state after. So in DNA, this can occur in like repair disorders, your double strand breaks, for example, DNA processing disorders. So like this is, division, kind of ties into the repair disorders, those kind of things. And then reduced expression of just general repair mechanisms. And for RNA dysregulation, and this is kind of like, transporting them, making sure they're degraded properly, blah, 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 that kind of stuff. And then the formation of aggregates, which I didn't know really until recently, that like if your RNA systems and repair systems aren't working right, your degradation systems aren't working right, then they can actually aggregate because with each other in certain areas. And so then that just becomes a problem you got to deal with. Because not only are they dysfunctional now, but they're kind of clogging up the cell. Yeah. Anything else? Cool.
Alrighty, so the inflammatory response is mechanism of defense of the body. It's part of the immune system. But as we were talking about in class, the brain has a blood barrier that though you do find T leukocytes in the brain, a lot of these functionalities are not being caused by your typical innate and adaptive immune system. So yes, T leukocytes are part of the adaptive immune system. I'm not saying they're not. But it's good to mention that inflammation is a defense mechanism of the body that causes the stimulus of a response to a specific thing that the body is recognizing as foreign. And so here there's like a silly little step-by-step. So it's going to recognize something harmful, it's going to remove something harmful, and it's going to stimulate itself to begin healing from the process of having that harmful thing. And so this can happen in the brain with the T leukocytes that we were talking about. This also mainly happens with glial cells. And through research, you can have when there's an overstimulus of inflammation through glial cells, you have these things called glial storms. And that's not really good for you. And this is also a hallmark of a couple different other autoimmune diseases or neurodegenerative diseases. And it works with other hallmarks to accelerate the process of neurodegeneration, of neuronal cell death. If you can think about a healthy neuron that's going to have this inflammation, like inflammatory response, and that, oh my God, sorry. Okay. And so if you're going to have these healthy neurons that are going to have these glial cells activated, Aaron, sorry. You're going to have these glial cells that are going to activate the inflammatory response. All of a sudden, you're going to have these microglia attacking their own neuronal cells from the network. And not only this is going to affect the functionality of those specific neurons, it's also going to affect the probability of a signal continuing through the neural network because of the affected neurons. And also that you're using a lot of resources with these glial cells that don't necessarily need to be activated because you're not actively fighting a specific antigen that's foreign. And so macrophages will be involved in this. Astrocytes will also be involved in this. And it just ultimately leads to a disruption in the neuronal network from functioning properly. Questions? Okay. So our last.
Oh, yeah.
So basically, the end result of all of these hallmarks and NDD properties is that we get neuronal cell death. So neurons are, like I kind of mentioned before, really high maintenance. So any sort of disruption to their normal state and their source of energy or function will kind of force them into cell death pretty quickly. And this is because one, it's highly specialized. It completes a relatively complicated process of transmitting info and electrical signals. But two, you have this cell body that sits here and then like extends all the way out, and then your synapses on the end and stuff. And so it's a generally big cell that requires a lot of maintenance to maintain. So you're with your gradual accumulation of macromolecular damage and your organelle damage, and the fact that you can't replicate, these cells don't self-divide. It's pretty vulnerable to that. High energy requirements, so any disruption to its energy source, ATP, flooding in, anything like that. If you don't have that, then it's just not going to work right. And like I said, extended axons and dendrites, and the cell's dependence on glial cells, and these glial cells kind of like maintain the environment and energy so that these cells can function. So if these cells start not getting a proper form of energy and the proteins and stuff needed to one, exist, or two, establish good relationships with these glial cells so that they can complete their own functions, then you're gonna die. That's just the end result. Yeah, and death kind of occurs in a lot of forms. It's not just apoptosis, it's can be necrosis too. And you guys know the difference, right? Like apoptosis controlled, necrosis not. It's kind of worse if you go that way, because then you're just like one, content spilling, not recycling, and kind of spreading out all over the place. Yeah, I think that covers our first, or I guess topic 2. You have any questions before we hit CML?
Wait, what was the uncontrolled one? You said apoptosis.
Necrosis.
Necrosis.
It's like programmed, this is not programmed.
Yeah. You kind of see that mostly in cancers. You want to read up on it?
Necrosis is worse because it's controlled.
I mean-
apoptosis is regulated, so it's like fine.
Yeah, like the cell knows it's going to die, and it completes a certain process before it does so. And I think pretty sure like apoptosis recycles a lot of the components of the cell.
And it's bad specifically on like diseases that affect mitochondria, because a lot of the apoptotic processes start with proteins that are regulated within either the mitochondrial matrix or around the mitochondrial cytochrome C.
Yeah.
And so if you have like malfunction in that process too, then that can also lead to like some pretty bad phenotypes for cancer.
I think the mitochondria releases that cytochrome C out of the mitochondria. Once it's out of the mitochondria, that's when the cell says, hey, it's time to apoptosis. So if you're getting like those from other cells because of some necrosis, then other cells will start to die.
So could you repeat why, like the structure of the neuron and like the axon and dendrites being larger than like the typical, I don't know, other body cells, worse, and like how it works?
Yeah, I mean, you're just basically it's a physical thing, right? You're relying on much more, let's say like skeletal features, extended like energy transmission, extended protein transmission. Like it's just these systems have to work in a larger setting, and it requires more energy.
Also, like axons, and like Schwann cells are structured with microtubules on the outside. And so you have regulatory like microtubule binding proteins like tau, for example, that regulate stability of the neuronal cell. And once these proteins, tau specifically, once it misfolds and aggregates and forms like neurofibrillate tangles, it leads to a lot of like neuronal cell death because of the lack of stability of those microtubules. So like microtubules by default are dynamically unstable because of just how the nature of microtubules is. And if you have these proteins that are there specifically to regulate the functionality of a specific microtubule, and all of a sudden they're misfolding and they're not performing the function, it makes it that much easier for the like the microtubule to just go into collapse.
And the fact that neurons kind of have to establish, which other cells kind of do too, but all neurons have to establish this like gradient between the membrane and itself, which is very important for its function. So like that's a not complicated, but consistent thing that needs to happen at all times. So if you get disruptions to that, you get lack of function and then death.
So we're gonna transition to CML. So chronic myelogenous leukemia. So hematopoiesis is the process of differentiation of blood cells. And this starts with two different lineages of cells. And so you're gonna have the myelogenous lineage and the lymphocytic lineage. I will say that specifically for this class, we will not focus on a couple of the specific differentiated cells of the myelogenous line. We will only be focusing on the important ones for the class. But it's worth mentioning that they do exist. And so the myelogenous lineage originates from the myelogenous stem cell, while the lymphocytic lineage originates from the lymphocytic stem cell. And so a couple of the, or not a couple, but like the lymphocytic stem cells will eventually differentiate into your B, T lymphocytes and also your natural killer cells. While the myelogenous lineage will differentiate into your leukocytes. It'll also differentiate into your platelets, into your granulocytes. And we'll go over that in just 1 second with this image. Again, like I was talking about earlier, on the myelogenous side, you do have more cells that we just did not focus on for this class specifically. And the cells that we will be focusing on are the erythrocytes, which are the regular red blood cells, the myeloblasts going onto your granulocytes of neutrophils, eosinophils, and basophils. So monocytes and macrophages do exist, as well as megakaryocytes and thrombocytes. We just will not be focusing on them. Any questions so far? No? Okay. So, you wanna do this?
Sure, that's quick. But chromosomal mutations can happen in a lot of forms, right? And this is a lot of review from 285, but like we have our insertions, deletions, duplications, inversions, but for CML, we're really looking at the chromosomal, that's a word, right? Chromosomal level. So like this is specifically the translocation, right? You have one part of a chromosome switch basically with another part of a chromosome, and they don't have to be the same size, right? Specifically in the CML, which is the 9 and 22 translocation, it's like a very short part of the 22 thing that like
We'll look over the here.
yeah, that like jumps over. And this is considered a balanced translocation event, and that just basically means that you're conserving DNA, right? DNA isn't lost, it's just in the wrong place, right? This isn't like a deletion where now you're like losing specific bases or anything like that. All your DNA is still there, but it's just in a different place. And that kind of helps. I mean, I don't know the exact reason or mechanism why, but I like to think that it helps keep the cell alive in a way. And like, that's why they can still exist in a cancerous state and not just instantly dies, because, you know, your DNA is still there. It's going to function. It's just not going to, now you have this fusion gene that's functioning weird, but it still works.
Okay, so I just wanted to outline a little bit of the molecular mechanisms. So we'll be covering domains, subunits, and modifications. We'll also be talking about ABL activation and inhibition, and BCR ABL1 chimeric gene formation. So domains and subunits, I've had discussions with Jen during review session because of this. But so domains are independently folded portions of a singular polypeptide, and they are highly evolutionarily conserved. A subunit is a singular 3D folded protein or polypeptide chain. And when multiple are present, it leads to the existence of, say it, Jen, I know you want to say it. Thank you, Jen, quaternary structure. Okay, I like to think about this as like the domain specifically, you can think about a single polypeptide chain being a train, and each of the individual wagons are their own domains. Okay, so a couple of the important domains that we'll be covering for this unit. The essay should know these domains, please know these domains, use these domains in answers. So SH2 domain, phosphotyrosine binding domain, SH3 domain. Anybody? Don't read. Okay, everybody read. Okay, proline rich region binding domain, and then PXXP motif. PXXP literally stands for proline, whatever, proline motif. And it means that it's a proline rich region, and evolutionarily, it will bind to an SH3 domain. What does SH stand for? Exactly. And why does it stand for sarcomology? Okay. Oh, well, I wanted to talk about important modifications that we'll be covering, but that's just the myristoylation I wanted to highlight really quickly. And so C-able tyrosine kinase, it is a gene found in human chromosome 9. It is a non-receptor tyrosine kinase present in the central cell pathways, such as RTK-MAPK or JAK-STAT5 or cell survival signaling. And so it is strongly regulated because of the importance in these pathways. So tyrosine kinases are commonly found as receptor molecules, but this is a free-flowing diffusing molecule that happens to rely on chance of running into substrate that it can phosphorylate. And it is also believed to be involved in DNA damage repair pathways, which means it can have access to the nucleus. I decided to not put this on the slides, but BCR is present on chromosome 22, and its true functionality by itself is not really known. It is believed to be a GTPase that acts around, or a GEF for the GTPase family of Rho, but you don't need to worry about that. So, able autoinhibition. So we spent a little bit of time on this in class. Oh, also stop me whenever you want. But so autoinhibition. This is going to start with the binding of a myristoyl group into its binding pocket in the cap domain. Upon this happening, hi Dasha. Upon this happening, we will have a conformational change that will lead the cap domain where the myristoyl is present to bind the kinase domain. And this, the docking of the myristoyl group into the kinase domain will happen. And so you will be having a bound cap onto the kinase. On this, there is another conformational shift, and it creates docking space for an SH2 domain to bind onto the kinase domain. And this happening causes a conformational shift in structure and position of alpha helices as the image shows. And so there's a 90-degree shift. With this 90-degree shift, you'll have a lot of proline present on those helical breaks. And so the SH3 domain will then be able to bind and cause autoinhibition of the able kinase. Is that clear? Too fast? Too fast? Okay, we can go back. So you have this very important molecule, this very important lipid called myristoyl. This myristoyl group is essential for the autoinhibition of the able kinase, and researchers were able to figure this out by checking the version of able that did not have the myristoyl binding domain, as able 1B from that paper. I forget what paper that is. Does somebody remember what paper that is? Yeah, and so once you have the binding of the myristoyl group to its binding site in the cap domain, that causes a conformational shift. What's a conformational shift? Is when the protein changes in structure after something stimulates it to. So this cap domain with the myristoyl group will then be able to bind the cap domain due to its conformational shift that just occurred. And with this happening, you'll be having the myristoyl group bound onto the kinase domain, leading the cap to be stuck to the kinase domain. This will cause another conformational shift due to binding, and it creates a docking space for an SH2 for the SH2 domain to bind next to the kinase domain. Once the SH2 domain is bound to the kinase domain, as the picture shows right there, then the conformational shift in structure happens again. And this causes a 90-degree rotation in the alpha helices. Once the alpha helices are shifted in 90 degrees, you'll have a lot of prolines because of the helical breaks that are present in between the different helices. As Aaron was talking about earlier, proteins happen to be in divided sections of secondary structure. And once those proline-rich regions are available, then the SH3 domain is gonna then dock, and it'll cause autoinhibition of Abel kinase. This is the wild type when BCR isn't there. Very important. Because Aaron is going to talk to us about the translocation event.
Alright, so we kind of already talked about this a little. But CML, this Philadelphia chromosome is what this 9 to 22 translocation event was named. And this was discovered in 1960 by Peter Noel at UPenn. I don't think you need to know that, but fun facts, I guess. And this is balanced. So like I said, DNA is conserved. You're just switching one part of this normal chromosome 9, which to normal chromosome 2, and notice it's like much bigger. But you get this large section from the 22 that jumps onto the 9, and this small section from the 9 that goes over to the 22. And that Philadelphia chromosome is on the 22nd one. This is where like that BCR Abel, and it's labeled pretty nice in different colors. That's where you get that fusion. And I guess this is a pretty perfect, like down to the nucleotide translocation, because you get this whole like BCR gene that's split, and then this Abel that like comes off. Oh, and something important. You see that little blue section of the Abel that's kind of left on? Yeah, that's left on. That kind of plays into why you lose that myristoyl binding, right? That cap domain that, you know, it's able to collect the myristoyl group isn't there anymore because we don't have that DNA. Instead, it's replaced by the BCR part, which we'll talk about in a sec, but that goes into playing into why you can dimerize and constantly activate. So yeah, so this translocation event leads to the chimeric formation of the BCR Abel 1 gene. And when it's expressed, this BCR Abel 1 fusion protein, like I said, is what exists. And like I just said too, it's that part that's left on from the translocation that's kind of stuck back on the 9th chromosome is what loses that N-terminal cap domain in ABL. So, you know, it's just downstream of effects, right? And, you know, it would, if you're taking the exam, it would be good to discuss like, you know, this leads to this, leads to this, leads to this, and this is why we end up with this. Like, it's good to talk about those things, but you lose that myristoyl group, you don't have binding to the SH2 pocket domain, right? If you don't have binding to the SH2 pocket domain, then that conformation where SH2 and SH3 are able to auto-inhibit doesn't occur. You don't get that curled shrimp formation. If you don't have that curled shrimp formation, it's going to be constantly active, right? That kinase domain is going to be available for activity for forever until you treat it.
Or would intrinsically goes into its inactive mode at some point.
At some point, yeah. Which is important for in that. We'll discuss that in a sec. Yeah, and so when multiple BCR Abel 1 proteins are near each other, dimerization can occur, and this primarily occurs because the BCR portion has an oligomerized, oh my God, words, oligomerization domain, right? It's the BCR part that's able to have that binding with the other fusion gene, and that's what kind of allows for the cross-phosphorylation to occur.
That's the N-terminal coil.
Yeah. Yeah. And so because they're in close proximity, cross-phosphorylation occurs and you get constant activity. Any questions? Cool.
Alrighty, so one of the papers references, it's the original paper, it references oncogenic addiction. And so this has to do with the overactivation and overuse of BCR Abel 1 kinase through the affected cells. And so it's been observed that in cell lines where BCR Abel 1 is present, that the cells start to overly depend on it. And this phenotypically describe, I got this from a paper. It phenotypically describes cell lines that are driven into apoptosis while the oncogene, in this case BCR-ABL, is absent. This isn't necessarily about ABL. This is just a paper on oncogenic addition of proteins. And so the oncogene becomes required to sustain the malignant phenotype, and so its absence, so if it cannot be activated, drives cell death. And so this is important because the authors talk about a couple of the different cell lines that have been observed to not continue to be oncogenic, and that is because once deactivation of the ABL kinase happens through the methods of treatment that we'll be talking about, then these cells are self-driving into apoptosis because they do not have the expression of the other kinases that they were using beforehand. And so it goes back to toxic loss of function and toxic gain of function. Because you're losing the functionality of the specific kinases that were performing other like cellular processes, and you're gaining the function of this overly active oncogene. In the case of BCR-ABL, and then the roles are reversed. Once you have the toxic loss of function of this BCR-ABL, then the cell that was overly reliant on it will drive itself into apoptosis. Any questions?
Is that directly linked with the certain people on the name of the protein? Like,
In general or for BCR-ABL?
like if you're saying like something has the toxic gain of function, would you be saying the gene has the gain of function or the protein?
The protein. But toxic gain of function has to do with the overly active phenotype that you're expressing / visualizing. It doesn't necessarily have to do with BCR-ABL. It's a good example in this case because you are like toxically gaining the functionality of like other kinases. It's specifically a tyrosine kinase. And so if there is an available phosphorylatable tyrosine that is terminal, then it can be activated / phosphorylated by BCR-ABL. So that's toxic gain.
So it's the kinase that has the toxic gain.
Like for BCR-ABL?
Yeah.
Well, you can call it the protein in general. Because it's the fusion gene. It's just, well, BCR-ABL by itself doesn't do anything if it doesn't have a kinase domain. But so the kinase activity of ABL is the functionality driving the malignancy of the leukemia, right? At least in the beginning, in the first stages. But it's a toxic gain of function. Okay, and so one of the papers mentioned this. BCR-ABL was the first malignant disease that was associated to cytogenetic abnormalities. So we were just going to cover quickly what cytogenetics are and why specifically this was the first one. So CML was the first one. This is because the translocation event was so specific and it led to such a specific phenotype of a chimeric protein that it led to the discovery of the Philadelphia chromosome on CML patients. However, it's important to note that Philadelphia chromosome positive is not necessarily only a CML characteristic. It has also been seen and observed in patients of acute lymphocytic leukemia that have the Philadelphia chromosome. Acute leukemias are different than chronic leukemias because they usually affect younger patients because of their less mature, more prominent to overproliferation, stem cells, and old, earlier, larger blast cells. And it is different from a chronic leukemia because, as opposite from what I just talked about, a chronic leukemia happens in the more differentiated, less theoretically dividing, older cells that are more mature. And so, why was it the first one again? Also because with a lot of other types of cancer, you need multiple mutations to occur in order to trigger something from happening. And when Aaron and I took this class over the summer, Laura used to like talking about how this was a perfect storm for mutations to occur, for you to drive cells into higher rates of transcription into higher rates of proliferation and into less apoptosis with the functionality of ABL and cell survival, and other very important pathways. And karyotypic analysis that I mentioned beforehand. So the left side is a set of human chromosomes. You have XY and XX there, but it's important to look at chromosomes 9 and 22. As you can see, chromosome 9 has a long arm or 2 long arms, and chromosome 22 happens to be shorter. The right side is the karyotype of a CML patient, and if you can look at chromosome 9 on the right side, the long arm is a little bit shorter, and the chromosome 22 leg or arm is a lot longer on the right side, which demonstrates the translocation event happening. So, diagnosis. You wanna take it?
Yeah, sure.
I can do fish if you want.
Why you want fish?
Whatever.
All right, so diagnosis, so there's a couple ways that the papers talk about, where we can kind of see if CML is going on. Most basic is your complete blood count. This is, just like a routine checkup exam where most CML cases are caught without, expectation. So this is pretty simple. I think it's like they take just like a small sample or look into a small sample like of a grid and they count like 20 cells or something. And they see if any of them are like, CML affected, because it's gonna be pretty obvious under microscope. And you'll generally see, and we'll talk about the phases, how this differs, but you'll see abnormal white blood count, blood cell counts. This is like 200,000 plus, and platelet counts, which is like 10,000 plus. Yeah, and so this is one benefit is that it's just super low cost, right? You can easily screen a lot of patients, and this is why it's generally like a first line, like, hey, this is a routine checkup, let's just see if everything's going accordingly. I think most of the time they're not even looking for this specifically, they're just checking if anything's wrong. But I did. Yeah, you're on your phone.
I think Haley is winning right now in Massachusetts. Right? Yeah. Anyways, back to not politics. So fluorescent in situ hybridization. So this is a fairly common method of checking for the expression of different proteins. It is a methodology that uses fluorescent probes attached to the end of mRNA sequences that are first permeated to be able to enter not only the cell but the nuclei of these cells. So not only the probes are permeated, but also the cells are permeated. And so you're able to target a specific sequence of an expressed mRNA. The mRNA would necessarily be in the cytosol, but you're also able to target DNA sequences that are complementary to the mRNA. And so in the picture right there, you have your red and your green probes that are respectively going, I don't want to say respectively, I don't know which one is targeting ABL, which one's targeting BCR. But the yellow dots, which are less commonly found, are the express sequence where the scientists specifically targeted BCR and ABL together. Laura did the demonstration of you going backwards on the mRNA sequence in order to get a sequence that matches both. And so they target the sequence that follows from one gene to the other to make sure that they're both in the same place. And that's why the probe shines yellow, because you have to have a different visualization method for any of the 3 targeted sequences that you're looking at. And so this is very useful because you're able to find where the protein is located in the cell, where a specific gene is going to be found in the chromosome. And it's just very good. It's not necessarily only used for CML. You can also think about other types of like cytogenetic mutations that you can look at. A common one is trisomy 21 that can also be targeted. And it's fairly commonly used as far as like the world of diagnoses go. Go. I don't know. Go. Oh. Alrighty, so jumping over to disease pathology. So we're going to talk about the different phases of the disease. And so we're going to start off in chronic phase. So this is the initial phase of CML. This is where a majority of your patients are going to be found, and it's where a lot of these patients that I just talked about will have no symptoms. And this is bad because you can have years and years of just having no idea that you have the condition, and that cells will be multiplying and that you'll have accruement and accumulation of mutations. And less than 10, very important, the numbers part, guys, very important. You're gonna have less than 10% of blasts in the blood and the bone marrow, and this is gonna be important for the symptoms that will follow up in the next phases. So yeah. Accelerated phase, so this is a more intermediate phase of CML. Here, 10 to 19% of your cells are going to be blasts. And as we were talking about, I'm gonna have to run back a couple slides. The less mature cells are gonna be larger in size than your more mature cells. And so if the cells that are being unable to replicate are your blast cells on the myelogenous line, then that means that those cells are gonna take up more space in the bone marrow than your more differentiated, smaller, more focused on specific functionality mature cells. So that would be your eosinophils, like all your granulocytes, but they take up less space than your myeloblasts. Let's go back. And so what's happening here is that those cells that are younger are able to replicate themselves, but they start becoming unable to differentiate into your more mature cells. And this is important because with time and with differentiation, they become smaller in size, and your bone marrow can take, in theory, the concentration of cells that is adequate for the amount of space that it has. And so because you have so many of these blast cells, what your bones do is they're like, oh my God, I cannot have this many cells in here. I have to ship these off somewhere else. And so you're going to have these cells be shipped off onto your spleen. And so what happens when you have these cells into the spleen is you're going to have an enlargement in spleen size. So that's where your splenomegaly comes from. And these cells are going to be so numerous and so unable to differentiate that they're going to start reproducing inside of the spleen, which will cause extramedullary hematopoiesis. What that means is hematopoiesis that is happening outside of your bone marrow. And so you're going to have spleen sizes that are going to be absolutely abnormal because you're going to have the expansion of the organ since there is the deposit of these cells. And you're also going to have the expansion of your bone marrow because not only it's shipping off these cells onto your spleen, but it's also having to like accumulate this larger number of blast cells that's present. With bone marrow expansion, you're going to have inevitably bone pain because your bones are not supposed to comport the amount of cells that they are trying to comport. And this is where resistance to Gleevec is going to start happening. And this is why the industry was forced to take an approach of the development of a second generation of tyrosine kinase inhibitors. We haven't just yet mentioned treatment on this review, and we will get to it in a second, but it's just important to know that starting an accelerated phase, Gleevec stops working. And here, you're gonna have a much quicker progression of the disease when compared to chronic phase. Yes, Dasha?
I just have a question, if they, do the cells, the blasts stop differentiating because they just accumulate too many mutations? And so things that are supposed to aid proliferation
Don't.
just stop working.
They are able to continue to proliferate within blasts, but they can't move on to promyeloblasts or metamyeloblasts or no, promyelocytes, metamyelocytes.
I was gonna add that kind of adds to their size too, right? Not only do blasts like generally bigger than differentiated cells, but they're cancer cells. So they're abnormally growing, hoarding resources, so they're generally gonna be bigger. And that kind of adds to that bone marrow expansion.
Yeah, and so this is gonna have a much quicker progression when compared to chronic phase. So in chronic phase, you can have a patient be asymptomatic for years and not even know that they have this condition. And when it's found out that they have the disease in chronic phase, then imatinib treatment works and it works very effectively. When you start getting to the more advanced stages, then Gleevec treatment stops working. If you want to get last.
Sure. So this is the most, I guess, deadly phase of CML that you can have. And it's generally met with a fatal prognosis. So this is when like greater than 20% of your cells are blasts, and the cells don't differentiate here either. The extramedullary hematopoiesis is a factor. Basically, it's too far gone.
right? These cells have so many mutations and now dependence on multiple you know, cancer sustaining pathways that just treating it with a TKI isn't going to be any good. Like the great part about being in the chronic phase and treating it with like imatinib was that, oh, we have this one dependence on a single fusion gene that's not working right. If we can combat that, we're good, we're golden. Once you get here, that the blast crisis, just like there's so much going on that even if you stop the BCR-ABL, the cell is like has so many other mechanisms that cancer can continue through. And same things, the bone marrow expansion, bone pain, all are a factor. And even second gen inhibitors are non-functional. I think really the only way for this is like chemo, but at that point it's too far gone. You only have this is only like 3 to 6 months. It's a pretty short period once you get there.
And they have the development of like third generation inhibitors, and if you look at the slides for like the second review session that we did on CML, you will see that they are effective in like treating BCR-ABL specifically, but at that stage, it's too far gone as far as the cancer prognosis goes.
Yeah. People with access, or like fortunate enough to have consistent access to healthcare generally get caught right here. So it's like 5 to 6 years in this chronic phase. So if you're not getting like a checkup where you're getting your blood tested or something, and you're not like, reporting any symptoms for 6 years, it's kind of hard to end up there, but it still happens.
And chronic phase is usually seen when patients are older than like 40 or 50 years old.
And CML as a whole kind of is generally affecting older populations only because it's not a heritable disease, right? It's purely like, you have DNA damage accumulating over time or from a specific event. And that's what causes the break. This isn't something that you're going to pass on to your kids. You may pass on like an inability or a dysfunction of like DNA repair mechanisms that may contribute over time to other like maybe cancerous things and like processes, but like specifically CML, you're not passing down to your children or anything like that. Yeah. Okay, so we can talk about treatment. I think that's one of the last things. I don't know why the fish slide made a reappearance.
Oh, I don't either.
It's very important, I guess.
Yeah.
All right. That's good. So imatinib, or Gleevec, whatever you want to call it, is your first generation wonder treatment. And we'll talk about if it's really a wonder treatment in a sec, but it was discovered in the 1980s by scientist team at Ciba-Geigy, or now Novartis, through a use of high throughput chemical compound library. So Laura briefly touched on this. This is basically just like mash up a bunch of chemicals and see what sticks and what works. And if they see something that works, they go further into that and try to figure out why it did. So there's really no I'm sure there's some rhyme or reason for like theory, like, oh, this might work because of this, but there's not like a designing like, oh, let me put this chemical group with this one and this. So like, it's a mix and mash. So the clinical trials pointed out that the absence of side effects from therapeutic usage had extremely positive results in trial. 92 to 98% of cancer cell lines were tested, presented complete cytogenic responses, according to the Charbonneau paper. And this is one very high number, but complete cytogenic response, we talked about this in class too. It's kind of vague. Like we don't really have a set definition of like what that means. But generally it's something good.
Well, they did have a set guideline for that, and it was the absence of BCR-ABL in 20 cells.
I think it's 18 F one.
Yeah, whatever
Like
it
that.
is, like that's just like infinitely too small to like be able to claim something like that. And Laura can go on her tangent about talking about the healthcare system and how they're not molecular biologists,
Yeah.
which I can agree with. But it's important to talk about here, and what she might ask you in the exam is that, you had that question in the problem set that talked about the complete cytogenic response and our ability to visualize and determine the prognosis of the disease. Let's make sure that we're talking about 20 cells not being enough, because that's just not enough, and we're not accurately able to determine such thing as a complete cytogenic response, specifically because you can also have just dormant cancerous cell lines that aren't active just yet, and that can activate over time. And if treatment has been ceased because of a complete cytogenic response and you have the activation of these cell lines, then it can lead on to new mutations and development, the relapse of the disease, even when it looked like a complete cytogenic response.
Good.
When they make complete cytogenic response, like I know in their like. Findings, it was like the 20 cells don't have it. Is that just what they meant? Is that our cancer, there are no cells, our bone marrow cells don't have. BCR-ABL? Is that what they mean by complete cytogenic response?
Well, if we think about like the oncogenic addiction part and we combo it with Gleevec being extremely effective, which it is. You can see that the cells that were like strictly dependent on the functionality of BCR-ABL and ABL kinase will drive themselves into apoptosis, right? It's a phenomenon that has been recorded and that you can visualize. However, that only acts with the activation of these other dormant cell lines, and you don't really have a way of claiming it. You just don't. If you're claiming it, you're just being like a bigot, but we don't have to talk about that.
Yeah, very ignorant, cause like, yeah, you're not ridding the body of those BCR-ABLs, right? It's still there, it's just dormant. You have your HSCs, which we'll talk about in a sec, like, it's that abnormality can still exist, and it probably does in those stem cells, but as far as those differentiated cells and what's actually causing the issues that lead to those like phenotypes of the enlarged bone marrow or the enlarged spleen or whatever, you're not gonna see that anymore because you're taking away the differentiated cells. But as far as like complete cytogenic response, no, it's definitely still there, just dormant. Yeah. So why does Gleevec work so well for cells that express BCR-ABL? And it does so because it works as a competitive allosteric inhibitor. And it's specific to the inactive conformation of this protein, and that's like we talked about it constantly being active, but there are points where it kind of returns to that inactive conformation and then re-enters the active conformation. Like it does have transition points every now and then, and that's when it's in that inactive conformation, that's when imatinib is assumed to bind, and it does so by binding right into that pocket where ATP would bind. And if you have that binding, you're not getting phosphorylation. If you don't have no phosphorylation, you have no signal, right? Pretty standard for TKIs. And over time, resistance can occur through a lot of mechanisms. One of them is a T315I mutation. This is John's favorite thing to talk about, is altering a threonine to an isoleucine. Yeah, you want to talk about why?
Yeah, so it's a common mutation, and I will draw. You'll have to enjoy my drawing. And the microphone will capture it, but it's okay, because you should have been at the review. So our group, well, I'm not gonna draw like the NCC, I don't, that's irrelevant. All we care about here is that you're gonna go from a CH3, OH, CH3 to a CH3, CH2, 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 CH2. Why is this important? So as we were talking about, and as Laura highly mentions in her other biology courses, when an enzyme or a protein binds a substrate, you don't want it to have the highest or the lowest dissociation constant in the world, because you need it to get rid of, you need it to just leave the binding pocket. So imatinib is going to bind onto the ATP binding site through non-covalent polar interactions, specifically where ATP would bind. And so if we think about position 315, right? If you're gonna go from a threonine, which is this amino acid that I drew right here, to an isoleucine, I'm not gonna write isoleucine. What are you changing here? What's the key change between these two, besides steric strain, doctor?
You're losing your hydroxyl group, so you can't form any hydrogen bonds anymore, and it's also a much bigger molecule. Isoleucine, there's a lot more carbon chains.
Yeah, exactly. And so if you're highly dependent on these nonpolar, interactions from imatinib to bind to the binding pocket, all of a sudden, when you have no hydroxyl group right here, as Dasha was talking about, you just cannot form your polar interactions, and that prevents imatinib from binding onto its binding site.
Yeah. Any questions?
Cool, discussion time. HSCs are often, so these stem cells are often left unaffected. Why? We talked about this a little in class, does anyone know?
HSC is.
Yeah, the hematopoietic stem cells. So these are the cells that like are at the top of the lineage and then we get differentiation down to the blast. We can, do you want me to go back to the lineage slide? Okay.
Do you want to answer, Doctor?
They think that inside these, in these stem cells, when you're going through, I think this is one thing, that we talked about in class. I don't know if this is the correct answer, the only answer, but they talked about that there are other kinases, present in these stem cells that are working, and so even if you are binding in, even if imatinib gets into the stem cells and is disabling BCR-ABL, the cells don't go into apoptosis because they have another thing, another kinase that they can depend on, and they don't die. And then when they differentiate, they lose that other kinase. I know that's something we discussed in class. I don't know if that's the like correct answer.
No, it pretty much covers it. You know, these stem cells work very different than the differentiated cells, right? They're not completely dependent on this BCR-ABL, and BCR-ABL expression is relatively lower, if not like non-existent in the stem cells. So you kind of don't see imatinib, you know, being that effective. So these cells are kind of like able to evade imatinib treatment and then continue to divide and make these effective differentiated cells. But you don't see that when you have imatinib because imatinib covers the differentiated cells. But once you stop imatinib, remission often occurs, right? Because you don't have that. You have an immediate relapse. That's the main problem with like the first gen, like you and a lot of the other gens, is that you have to take it forever. You can limit the things that cause the major problems, which is those differentiated cells, but you're not gonna hit the root, which kind of sucks. I know there are treatments that you can where you can do like stem cell transplants, but generally, since this affects older patients, it's kind of hard, you know. One, it's costly, and two, it's like taking that risk to do that stem cell transplant that might be rejected, or you know, because these are older patients, they can't handle it. It's just like kind of useless to go that route. But in theory, it would work.
Can you just go back a slide?
Yeah.
Yeah, so I wanted to talk about the method of inactivation or like inhibition through imatinib. Imatinib only works, again, I want to emphasize that inactive conformation. And just to go back to like the entering the inactive like state that we were talking about earlier, we don't really know how this is happening. We just know it happens. So it's just one of those things that like we intrinsically know goes into its inactive state, even with dimerization and tetramerization of the BCR-ABL. And that's why in higher quantities, imatinib is able to perform its inhibitory mechanism. And so it's just important to clarify that if you're asked in the exam about the functionality of imatinib, don't talk about an active inhibition of the molecule through imatinib, because it's only occurring when these tyrosine kinases are on their inactive state. I just wanted to like emphasize that.
The future generation treatments are targeting the active.
Yeah, but at that point, you're basically like targeting that for like no avail. Yeah.
Oh, and one more thing, this wasn't in the papers, but I kind of read it, which is cool, is that imatinib can force the stem cells to increase expression of a certain cell migration factor. And it forces the stem cells to actually like migrate back into the bone marrow, or like kind of get tucked away and kind of like literally hide from imatinib. It's cool. None of your papers talks about this, but it's just a cool fact of how it also evades imatinib response.
I mean, it's good they're going back into the bone marrow.
In a way, yeah. I mean, it's they're hiding for sure, but like they're still there. So anytime if you stop imatinib, they're gonna come back out and kind of like, you know, go into a position where they can continue to differentiate.
Yeah, and this is just a brief overview of like second generation inhibitors. So these are also small molecule inhibitors that were developed with mutation in mind. And so a couple of the common ones are the phenylalanine for leucine, and also the tyrosine 253 for phenylalanine mutations. You don't need to know these mutations by heart. It's just to provide information about how the development of these drugs was happening. And they're there in order to combat inefficiency of Gleevec in treatment and try to prevent the activity of BCR-ABL. But as we were talking about, second generation inhibitors only start working once patients are like in accelerated phase and moving on to blast crisis. And at that point, as Aaron and I were talking about earlier, you just accumulated a way too great number of mutations in order for this to have any major causal factor. And the major difference between the first generation and second generation inhibitors is that Gleevec is actively able to prevent the prognosis of cancer because of its important functionality in the cells and because of BCR-ABL being the main cause of these the overproliferation of these leukemic cells. But when you get to the stage of the second generation inhibitors, it's just very much too far gone. You are actively inhibiting the functionality of BCR-ABL, but at that stage, there are many other mutations that cannot be taken care of. So, yeah, so dasatinib, nilotinib, sunitinib, and there are less survival benefits when compared to Gleevec. And just to touch on combination therapy, so the use of TKIs with chemo can be used to overcome resistance. Why do we think this is the case? A cold call. Grace? Use of tyrosine kinase inhibitors with chemotherapy can be used to overcome resistance. Why do we think this is the case? Carla?
They have like a synergistic effect where that you can like target the mutation and then chemotherapy comes like.
Yeah, an example that I like using, and it goes back to me naturally because 285. Laura spends a little bit of time talking about Taxol being microtubule stabilizers. So if there are the spindle formations happening and you have chromosomes being pulled apart in the spindle, and you're freezing or stabilizing microtubules with Taxol, what's going to happen? If the cell is in a stable microtubule state for too long, it drives itself into apoptosis. And so if you are inhibiting BCR-ABL from having like very active conformation in like early stage CML, and you are also using combination therapy with chemotherapy drugs such as Taxol, then you can have the effect of the tyrosine kinase inhibitor preventing the proliferation of the cells, as well as the chemotherapy drugs driving the cells into apoptosis. But this doesn't work for like every case, and there are certain limitations that we have to deal with. And so what do we think are some of these limits that we have to be worried about for this combination therapy? Dasha?
Just a clarifying question, is chemo the one that targets all proliferating cells?
The actively proliferating
Proliferating
cells,
cells.
yes.
So I guess that's kind of.
Which is why a lot of money is wasted in cancer research, because we are very good at targeting the actively proliferating cells, but we can't target the dormant cell lines. And then they are the ones that ultimately go back into activity and then cause the patients to go into a relapse.
That's specific to Taxol, right? So like, there are other chemos that can like.
I would mention, I think Taxol is the easiest one to talk about. It has the easiest functionality, and it just, like, the fact that the cells are stuck in the spindle assembly makes it so that they drive themselves into apoptosis. It's like a natural reaction.
Especially considering like the drawback of the TKIs, especially imatinib that we talked about, was evasion of the stem cells, which are actively proliferating and dividing, right? That this is where chemo might be effective.
But why do we think what are some of the limits that we have to be wary about with combination techniques? Yeah.
I mean, like chemo kills all actively proliferating cells, and at some point, it's like, what's killing the patient more? Is it the cancer or the chemo? And so that's just an element of chemotherapy, why everyone's looking for other possibilities. So I guess it's just kind of a race to see I guess that's the limit, but.
No, that's exactly what we were targeting with that. That's exactly it. So, as Sasha was just talking about, if you're actively targeting every proliferating cell in your body, then you're not only gonna have the successful target towards the proliferating BCR-able positive cells, also going to be targeting every single one of your proliferating cells in your body. And we can think about ways of, tissue regeneration or like tissue proliferation or literally anything that involves cellular regulation of proliferation being completely wiped because of chemotherapy, then that is a very, very active limit of what we can do in combination with these therapies. Any questions? Yeah,
At what stage in CML would,
any stage. It's harder to catch chronic phase because you just don't know you have it. But if the patient is doing routine checkups, like with complete blood counts or full blood counts or whatever you want to call them, this can also be an option because it's like chemotherapy is with radiography are the two most common ways of targeted cancer therapies. But if you're able to target a specific, because imatinib is just extremely specific, and it's also another one of the questions that you had in your problem sets was why does imatinib work for this but not for anything else? And it's specific enough to the formation of the chimeric gene, being the causal factor of the leukemia that you're able to use imatinib. But with any other cancer, there are so many other causal factors that work together in order for you to have like a cancerous phenotype.
Which is what we see in like the accelerated phase of even CML, right?
Yeah, it's starting at accelerated phase, but it's different. So the major difference between like leukemia and any other cells is that leukemias are unable to form tumors because they are in plasmatic environments, right? The blood. And so there's no like physical tissue like embodiment of a leukemic tumor. Like leukemic tumors don't exist. Whereas you have benign or malignant tumors elsewhere that form, and then the cells are able to go elsewhere because of that culmination into like the formation of tissue. Yeah. Any questions? No. Okay. So that will be all for tonight. Thank you all for coming. We'll be staying after for questions as usual. But the video will be up on YouTube hopefully soon. And I'll be sending the slides out also soon. But yeah, thanks for coming.